Okay, just one last thing about lipids. You know, the key things we have identified, the key structures, hydrophobic, hydrophilic, all that good stuff. The one last thing, other than biological membranes, other than energy source, other than insulation, a couple other key functions of lipids are steroids with cortisol and testosterone. These are steroids. They're anabolic steroids actually have the ability to penetrate the cell membrane because they are lipids. So steroids, the key thing, we'll talk a lot about steroids when we talk about the endocrine system. They can be components of vitamins, such as vitamin D. That's just knowing that they are components, nothing too big there. But the main one here, the other big one here is cholesterol. Cholesterol is a very key lipid. We think of cholesterol as being bad because cholesterol in your bloodstream can cause blockages and it can cause some type of cardiovascular diseases. But cholesterol is vital. Remember when we mentioned the lipid bilayer? The phospholipid bilayer has the unsaturated fatty acid tails. So your cell membrane is actually very fluid. Well, we don't want it to be liquid. We want to solidify it a little bit. We do it with proteins, but we also do it with cholesterol. You make cholesterol in your cells in order for that cholesterol to get into your membrane to help solidify your lipid bilayer. So just a couple more things on lipids, some side notes. And then the last one that's mentioned is lipids can be components of pigments. Carotenoids are a class of lipids involved with pigments, colors. When you see the leaves changing, you see the yellows and the reds. Those pigments are carotenoids, and they're lipid-based molecules. Not to memorize structures or anything like that, but just to be familiar with some other functions of lipids. All right. The focus of today are proteins. Proteins, they're the heavy lifters in our cells. Right? We are walking chemical reactions, and what regulates these chemical reactions are biological catalysts, which are enzymes. So some key functions of enzymes. They can be structural support of proteins, such as muscles, bones. Proteins play a role there. They can be part of protection. So they can be key components in your immune system, such as antibodies are protein-based molecules. So antibodies. They can be part of transport, which is, you know about these transport proteins in your cell membrane, osmosis, diffusion, things like that, facilitated diffusion. Catalysis is just simply saying enzymes. That's the main function. When you think of proteins, we just think of enzymes. Defense is just referring to our immune system again. Defense as such as antibodies, protecting us from certain pathogens. Regulation, proteins can help regulate homeostasis. If you think of the production of insulin, that's an enzyme-regulated production. If you think about the production of other molecules, the enzymes play a role in regulating chemical reactions. And then movement, pretty straightforward, right? You think of movement with muscles, but also just movements within the cell. There are proteins known as carrier proteins. So all of these are key functions of proteins. If you're going to boil it down to one or two that we want to be familiar with, the main one here is catalysis. They're enzymes. They regulate chemical reactions. And they're also involved with movement as well. So this is just a list. You can kind of read through these on your own, just giving you some examples and relating it to types of proteins and what they are. So you can kind of look at this list on your own. All right, so what are proteins? We have the monomer form of a protein, which are amino acids. There are only 20 different amino acids out there. That's pretty amazing, right? You would think how complex our proteins are. There must be hundreds, if not thousands of amino acids. There's not. There's only 20. 9 of them are essential, which means 9 of them we do not have the ability to synthesize or to make within our body, so we must consume them. The other 11, we can make them in our body. Yes.
The ones that are made in our bodies, is it from bacteria in the gut, or is it just like water?
I'm not sure. I'd have to look at each one individually. Yeah, I have to look up. I don't know. I'm not quite sure. I mean, we have the ability to formulate some ourselves, but there could be some that might be formulated by bacteria. So I'd have to look that one up. Yeah. So we have amino acids. These amino acids join together to form the polymer forms, which are polypeptides, right? Long chains of proteins of amino acids. So here's a nice little space fill molecule here. Okay. When we look at proteins, we need to break down the amino acid. Here's where our functional groups start coming in order to recognize. Amino acids have two key functional groups. This one here is dissolved in water. It's in an aqueous solution because the carboxyl group has lost its hydrogen and the amino group has picked up that hydrogen. So it's in aqueous solution. You also see this as NH2 or COO with an H. They got that minus right there. So they're the same thing, but that one's dissolved. That's why it's showing an acid. Okay, so key things. We need to be able to recognize the amino group, our carboxyl group. We have our central carbon, but we also have this R group. We have 20 different amino acids. It means we have 20 different R groups. R group's the variable, just like in X in algebra. So pretty straightforward. We're just going to key structures of the amino acid. Again, being able to identify the key functional units is something we need to know. Our amino acids join together via dehydration synthesis. Right, we have two amino acids come together, dehydration synthesis. A bond is formed, a covalent bond, which is called a polypeptide. We call it a peptide bond. The peptide bond is formed, and in this case, bless you. In this case, bless you. Woo. In this case here, we have a dipeptide. A dipeptide. If I were to break it apart, we would undergo the process of hydrolysis. So here are your 20 different amino acids with their R groups. Again, for each one, don't be confused by the R groups. You can still identify the amine and the carboxyl sections of each one. The blue is the functional group, and just kind of highlighting some key things. Some of these functional groups form acids in solution. Some of these functional groups form bases in solution. Some of these functional groups are non-polar. Some of these functional groups are polar. Some of these functional groups have sulfur atoms or disulfide bridges, which we'll talk about. But it comes down to it's the interaction of these functional groups that dictates the structure of your protein, of your amino acid. And the structure of your protein, right, of your enzyme possibly, dictates the function of that enzyme. So you can kind of get a look here. Here's another one. So we're just looking at some non-polar ones here. You can see there's, we don't really find any oxygens up there, right? There we have some carbons, some hydrogens for the most part, and they're going to be non-polar ones, which are going to try to stick away from water, so embedded in proteins, embedded in cell membranes. Yes?
It's like the carboxyl group, so like those sections.
Yeah, but they're going to be bound, right? So they're going to be, so this is not going to be off on its own. There's going to be a peptide bond, which is going to cut down on its interaction. But yes, I mean, if I have this oxygen group still there, there's still going to be some polarity there. But the interaction is going to happen more with the other group. But there will be some. Here are some of our polar ones, right? Here's some of our electric charges. Some are acidic, and some are basic, basically forming negative charges versus positive charges in solution. You don't need to memorize any of these R groups. You don't need to do that. I'm showing you these just so you can start seeing the interaction. When I have my polypeptide, we get these complex structures, right? We get these three-dimensional structures. They're not random. As much as that looks random, it is not random. It is dictated by the R groups, by the interactions. It's dictated by polar R groups trying to embed themselves into the middle of an enzyme so they can stay away from the water. Sorry, non-polar R groups embedding themselves into the middle of an enzyme trying to stay away from the fluid on the outside. Okay? And then the polar R groups are trying to be on the outside of the enzyme so they can interact with the water. It's the positives being attracted to the negatives. causing folding in specific ways. So it looks very random. It's not. It is not random. This just kind of gives you a couple of key things here. The N-terminus is the end of the polypeptide chain that ends with the amine group and nitrogen. The C-terminus is the end of the polypeptide chain that ends with the carboxyl. So one side they have an amine, the other side of the polypeptide they have a carboxyl. Okay. When we talk about proteins, we really talk about 4 levels of protein structure: primary, secondary, tertiary, and quaternary. And what we have here is primary is pretty straightforward. It is simply just the sequence of amino acids. So I can analyze a protein basically by looking at the genetic sequence, and I know that I'm going to have leucine, isoleucine, glutamic acid, and that's going to be the sequence of amino acids. So I can look at amino acids directly from the sequence of amino acids. Pretty straightforward. That's the primary structure. The next thing that's going to happen, just like with the glycosidic linkages, they can have alpha bonds versus beta bonds, depending on what direction they're facing. The same thing happens with amino acids. Amino acids can form certain bonds that cause them to spiral, which is our alpha helix, where we have alpha bonds causing our amino acids to form a spiraling type shape or a helical shape, a helix. If the amino acids are bonding in opposite directions, you can get what's known as a beta bond. We get a pleated sheet type shape. These are secondary structures. So real simple. Primary is the amino acids. Secondary just either alpha helices or beta pleated sheets. Those are the only examples we have. One or the other. They're either bonding straight up or they're bonding opposite one another. Alpha, beta. All right. Now we start looking at tertiary and quaternary structure. So here's tertiary structure. I like this picture because it's showing you I have a long chain, a long polypeptide chain that's folding, and within the chain, it's showing examples of beta pleated sheets here, alpha helices here. Like they're all part of the polypeptide. So they're there. Something's going on here too. This is either an alpha helices or a beta pleated sheet as well. So it's all over the place. So tertiary starts to show this 3-dimensional structure, this globular structure, but it is a single polypeptide chain. Notice, the structure looks random. It's not random. It's folding this way because of the interactions of the R groups. And there are specific interactions that are going to hold it together, such as polar, non-polar forces, and van der Waals forces, which I'll read what they are in a second. Again, this is showing a disulfide bridge. So some of our amino acids have sulfur in the R group, and when those 2 sulfur R groups can get close to one another, they can form a covalent bond, known as a disulfide bridge. So all of these interactions hold together. Quaternary structure looks very similar, except with quaternary structure, there's a different shading here. I don't just have one polypeptide chain, I have numerous polypeptide chains, 2, 3, 4, as many as you can have. Right? So they're both forming similar 3-dimensional structures, but now we have multiple polypeptide chains joining together, being held together by those interactions. Van der Waals forces, hydrogen bonds, and so forth. So this is just giving you a list of kind of the forces that help to hold tertiary structure. So van der Waals forces are simply molecular forces between molecules. So I have 2 separate molecules that come close to one another. Even if those molecules are non-polar molecules, they will interact with one another. Electrons will push on each other, and a region of one molecule could become a little bit negative, because electrons might kind of aggregate there. And the molecule it's coming close to, because they interacted, is going to become a little bit positive. And there'll be little attractions there that are constantly shifting and moving around all over the place. So that's just kind of a little interaction between chemicals. We mentioned the disulfide bonds. If I go back real quick to our list, the cysteine right here, we see that sulfur and cysteine. When 2 cysteines come close to one another, that's where you get the disulfide bonds. So it's really unique to our cysteine right there. Okay. Aggregation of hydrophobic side chains. That's just saying I have a series of amino acids that their R groups are non-polar, right? They're hydrophobic. So I get a series of amino acids where all their R groups are non-polar, right? They're going to force folding into the center of that protein most likely. They're going to try to avoid connections. They can form van der Waals versus ionic bonds. They literally can form ionic bonds with one another. And then hydrogen bonds, we should be familiar with from prior. But when you get a polar molecule coming close to another polar molecule, you can get an interaction there known as a hydrogen bond. It is not a covalent bond, it's not an ionic bond, but it is fairly strong. And then there are similar interactions with quartet. You do not need to differentiate between the two. It's not like you need to know which ones are tertiary interactions, which ones are quaternary. They cross over, some work, some don't. But just know that these interactions are holding together. Side note. So again, something that you guys can play with. There's an online game known as Foldit. Anybody ever heard of Foldit before? I think you just Google it, Foldit, F-O-L-D-I-T. And what it is it gives you sequences of amino acids. And it's the person's job to play the game to try to fold the protein into its appropriate shape depending on the R groups. When you play this game, you're actually working on specific amino acid sequences. And folding amino acids or folding proteins is not an easy job. Some of the foremost scientists in the world spend years and years and years trying to figure out the exact structure of one particular protein. So it's not an easy thing. Even supercomputers, because these interactions are subtle and there's variations, even supercomputers have problems running data trying to figure out from an amino acid sequence what the tertiary quaternary structures will look like. In this game, the person who's, at least a few years ago, the person who is best at this game isn't somebody from Yale, isn't somebody from Harvard, it's actually someone in my class as well. It's actually a lady who is a housewife from northern England. And she would just, in her spare time, start folding proteins on Foldit.com. So she actually was the best protein folder in the world. Was just this random lady who decided to play the game, have to be good at it. Just kind of interesting, just a side note. Okay. She recognized. I don't know. And you can go to Foldit.com. You can play with it right there. You'll see the rankings of people playing the game. Okay. So just a couple quick things. That's it. That's all we really need for proteins. We need to know that they're made of amino acids. They're polypeptides. They have different levels of structure: primary, secondary, tertiary, quaternary, and those structures are unique, and they're not random. Right? They're dictated by the R groups. What happens when we get a mistake? What happens when your DNA has a mutation, right? And instead of having a certain amino acid out of position, there's a mutation that causes a different amino acid. Well, sickle cell disease is a good example of that. It's kind of hard to see here, but it's giving us our sequence of amino acids: valine, histidine, leucine, threonine, proline, and then we have glutamic acid. So here you should have glutamic acid, but there was a mutation in the person's DNA. The DNA dictates the amino acid, and the mutation caused a valine to be substituted. Sickle cell anemia deals with your red blood cells. In particular, it deals with the protein in your blood cells that carries oxygen. What's the protein in your red blood cells that carries oxygen?
Hemoglobin.
Hemoglobin. So it deals with our hemoglobin molecule. People who have sickle cell disease have a mutation. That valine is substituted. And one of the subunits, which is a component of one of the polypeptide chains forming our hemoglobin protein, our 1, 2, 3, 4, our quaternary structure, is mutating. So this beta unit right there changes its shape. And when it changes its shape, it starts to have weird interactions with other hemoglobin molecules. So instead of having normal interactions in picking up oxygen, releasing oxygen, it starts to kind of form these kind of crystalline structures within your red blood cells, which causes your red blood cells to change from their standard circular shape or disc-like shape into kind of this cyclic shape. Not only is it signal, is its shape different, its ability to carry oxygen is different as well. Just kind of showing, hey, guess what? A change of a single amino acid could have a huge effect on the function of an entire enzyme or protein. Here's the other thing though. Major changes in amino acids sometimes have no effect too. So an effect doesn't mean it's going to be negative, it doesn't mean it's going to be positive. It depends on exactly where it is. So if I picture this, and I'm looking at this enzyme right here, if I have a major mutation that causes changes to the hydrophobic R group sequences in the middle of it, there's a chance it might have no effect on the enzyme. Enzyme might still operate completely 100% normal. However, if these sites here, this spot right there and those spot right there, if those are the active sites where the enzyme's actually working, if the mutation happens right there, it could be 100% non-functional. So just kind of understanding that, hey, a single amino acid could completely shut down the functioning of an enzyme. However, just because you have a mutation doesn't mean it's going to. Where that mutation is important as well. All right, two quick things and we're done. Protein folding, just an interesting term. This is nothing big, but a chaperone is just kind of a fun little protein that's in our bodies. It helps with protein folding. Again, when they fold, they make these fairly crazy three-dimensional structures. They're complex, they're delicate. Sometimes they need to be aided in their folding. Sometimes, in order to fold 100% properly, it might need a slightly different environment. Maybe it needs a pH that's a little bit lower than inside your standard cell, or a little bit higher. Maybe we need less or more salt content. All of those things affect the folding of a protein. And we have these other protein dividers known as chaperonins. Kind of a cool name. And what they are is the polypeptide we're trying to fold can go into the chaperonin in its own little microclimate, right? So a little bit more, a little higher, a little more acidic, a little less acidic, who knows what it is, and helps to fold it to its proper shape. But one last thing with proteins, just knowing what happens, right? It's why if you get a fever of 104, 105, 106, you should go to the doctors. Why do you need to go to the doctors when you get a fever of 105, 106? Because your proteins start to denature. These three-dimensional structures, they are delicate. When I increase temperature, I increase the movement of molecules. They start vibrating more. And if they start vibrating too much, the proteins holding the three-dimensional structure start to break down. The hydrogen bonds, the ionic bonds, the sulfide bridges, they start breaking. And your proteins start unfolding. An enzyme that unfolds is no longer working. So if you get a fever, 102, 103, for a long period of time, you got to be a little worried. You don't want to have that for more than a day or two. But if you get like 105, 106, you need to go to the doctor right away because your proteins really do start denaturing. Denaturing is not just temperature. We also have pH. Right? Increases or decreases in pH could cause protein denaturing as well. And we'll do a whole lab working on that. That's it. That's all we have for today.